Good day, this is Group 25 under the field of medicine. Efficacy of antibacterial activity of sanitizer spray derived from Coleus amboynicus or oregano. Background of the study. Coronavirus disease or COVID-19 is an infectious disease caused by a newly discovered coronavirus. Sanitization is a preventive and strategic method to contain spread of SARS-CoV-2. The best way to kill germs is by washing hands with soap and water. However, soap and water is not always accessible. So the recent pandemic has caused a panic buying of sanitizer and other disinfectants. Using hand sanitizer prevents the hand transmission of germs. A study by Simergiotis et al. 2020 stated that oregano or Oreganum vulgare is an aromatic plant with a wide distribution throughout the Mediterranean area and Asia. According to another study by L. Enciasi 2016, the antibacterial and antioxidant properties of oregano have been attributed mainly to carvacrol and thymol. This study aims to determine the efficacy of antibacterial activity of sanitizer spray derived from Coleus amboynicus or oregano. Statement of the problem. This study generally aims to know the efficacy of antibacterial activity of sanitizer spray derived from Coleus amboynicus or oregano. Specifically, this study aims to answer the questions: first, what are the antibacterial properties present in Coleus amboynicus or oregano? Second, what is the effect in the presence of bacteria of the component carvacrol and thymol? Third, what are the impacts of organic sanitizer in terms of healthcare and expenses? Fourth, what is the effectiveness of the Coleus amboynicus or oregano for antibacterial uses? Hypothesis. Coleus amboynicus or oregano has antibacterial activity.
Significance of the study. This study is for better understanding of the effectiveness of oregano or Coleus amboynicus extract using the improvised petri dishes for bacterial development, which will be beneficial to the following students, because this study may give information and better knowledge about the benefits of oregano as an antibacterial. Teachers. Teachers may use this study as a basis in teaching medical or biology subject. Community, because this will enable a community to widen their knowledge, especially to the people in the community suffering in pandemic due to spreading of coronavirus disease. Future healthcare practitioners, to gain new knowledge regarding the natural production of disinfectant. Future researchers, through this study, it will serve as a reference, material, and guide for next researchers in order to gain additional information about the efficacy of antibacterial activity of sanitizer spray derived from oregano. For the scope and limitation, this study was only focused on the efficacy of antibacterial activity of sanitizer spray derived from Coleus amboynicus or oregano. This study was conducted at the researcher's residence on the date of March to June 2021. Any further study that is not involved to this study will not be included.
Methodology.
For the research design, the purpose of this study is to determine the efficacy of antibacterial activity of sanitizer spray derived from Coleus amboynicus. In this study, the researchers use experimental study as the research design under quantitative method. It is also a quantitative research method such that it will describe the antibacterial components of the Coleus amboynicus. Materials that were used in the process of extracting Coleus amboynicus are oregano, vials, measuring cups, strainer, stirring rod, filter paper, funnel, spoon, graduated cylinder, droppers or syringe.
General procedure. The procedure for production of Coleus amboynicus or oregano pure extract includes gathering, washing, and squeezing of the raw material, then filtrating and storing of the extract. The procedure for production of Coleus amboinicus or oregano extract in boiling process is gathering, washing, and boiling of the raw material, then filtrating and storing of the extract. While the procedure for production of Coleus amboinicus or oregano extract in pulverization process includes gathering, washing, drying, and pulverizing of the raw material, then storing of the extract.
The experimental sanitizer spray. After collecting samples, the researchers provided 27 plates. The three processes of extraction, the pure extraction, boiling method, and pulverization, have 9 plates each, with 3 setups with 3 trials. Each plate consisted of 4 paper discs with a total of 108 paper discs for the percentage of bacteria data gathering. The following were the setups for each treatment. Set A contains 0.2 cubic centimeter or 100% of the extract. Set B contains 0.10 cubic centimeter or 50% of the extract and 50% of the solvent, while Set C contains only water or 100% of the solvent.
Preparation of agar plates. The agar plate is a petri dish that contains a growth medium solidified with agar. It is standard solid support material for growing microorganisms. There were 3 processes of extractions: the pure extraction, boiling method, and the pulverization. Every process have 3 setups, and each of them have 3 trials for precise results. The petri dishes have lid for storing to avoid contamination. The agar plates were contained a mixture of agar and water and stored to keep the media in semi-solid state. After pouring in the containers, the plates were stored in the refrigerator for several hours.
Storing of samples. After collecting the samples, they were processed and incubated at a room temperature. The researchers conducted an observation each day, and the samples were returned to its original location after monitoring until the last day of observation.
Observation of samples. The researchers performed a physical test based on bacterial growth in each of the sample to conduct qualitative analysis for observation. The samples in each agar plates were observed based on its physical appearance. The size, shape, and color of colonies present in each agar plate were observed during the descriptive analysis. The researchers conducted an observation every 24 hours prior to the storing of each samples. After each observation, the samples were returned and left to its original location of storing for further observations. The observation lasted for approximately 168 hours for the data collected.
Percentage of bacteria present in the agar plate. The percentage of the amount of bacteria present in the agar plates were calculated through a table and graphing of the samples. The growth of bacteria was recorded by the number of paper discs with the bacteria visibility that was applied to each plate, divided to the total number of all paper discs, then multiplied by 100.
Results and discussion.
Number of paper discs with visible bacteria per agar plates. Table 2.1, 2.2, and 2.3 shows the effects of the different setups of oregano extract on the paper disc located per agar plates after storing in 7 consecutive days. It shows that treatment C, setup A, shows the most effective in preventing bacteria growth.
The chi-square test goodness of fit. Antibacterial activity test in different processes: pure extraction, boiling, and pulverizing. Based on the result of the antibacterial activity test in pure extraction, boiling, and pulverizing of oregano leaves, the researchers wanted to get the effectiveness of the treatment to produce natural antibacterial sanitizer spray. The researcher used 27 samples and the effectiveness of each variables. Treatment A, B, C specifically was classified not effective, somewhat effective, and very effective. The scheme included a 95% chance of acceptance and 5% chance of rejection. Table results. The table shows that 7 of the treatment setups were not effective, 10 of the treatment setups were somewhat effective, and 10 of the treatment setups were very effective. The computation using chi-test is listing the observed and expected counts of our samples. Using the formula observed minus expected square, we found the critical value of x squared, which is 24.96. The degrees of freedom were calculated as n-1 since the value of n is 3 due to 3 rows. The degrees of freedom is 2. The level of significance is alpha 0.05. The critical value is greater than 5.991, therefore the hypothesis Collius amboinicus or oregano has antibacterial activity is accepted. Conclusions. In the course of the study, the researcher was able to accumulate and collect all necessary information. Therefore, conclude the following. The process of extracting antibacterial property from Collius amboinicus or oregano leaves has a significant impact on the effectiveness of the obtained product. Therefore, there is significant relationship between the different processes and the effectiveness of the obtained antibacterial property from Collius amboinicus or oregano. Setup A of treatment C, extract from pulverization of Collius amboinicus or oregano leaves, is the most effective process of obtaining antibacterial property from Collius amboinicus or oregano. The extract from Collius amboinicus or oregano leaves showed high antibacterial activity or is effective to prevent growth of the bacteria.
For the recommendations, based on the findings and conclusions obtained, the researchers offer the following recommendations for further study. 1. Lengthening the period for trial and observation required in order to acquire more reliable data on the product's effectiveness. Number 2. To strengthen the research and experimentation, the researchers should include more studies. Number 3. To guarantee the product's effectiveness, conduct an approved laboratory test with authorized laboratory personnel.